Well, triple-negative breast cancer indeed is a big challenge for us as clinicians because it accounts for approximately 15% of all cases of breast cancer, and unfortunately is characterized by high level of clinical aggressiveness and lack of a molecular target that we can focus on for obtaining clinically relevant results. The triple-negative breast cancer has not the estrogen receptor, the progesterone receptor has not the HER2 receptor. It means that the only weapon that so far is available is chemotherapy. And with chemotherapy we have some good results sometimes, but unfortunately most of the times we have disappointing results, either because the tumor doesn't respond at all to the treatment or because the response is very limited in time, and after a few months from the time of response, the disease is still progressing. So we certainly need new ways for attacking and controlling this type of disease. We feel that probably one of the most promising areas is targeting the incapacity of most triple-negative breast cancer in repairing the DNA. But we realize that this perhaps is going to work only in a minority of triple-negative breast cancers, because as you were mentioning, the main issue is that we have now quite clear that triple-negative breast cancer is not one single disease, but it's a sum of different diseases. And so we are quite sure that if we want to be successful in this setting, in this field, we will need first to characterize the different subtypes of triple-negative breast cancer and then to target each subtype according to the most biologically driven and clinically relevant strategy. PARP inhibitors are a promising drug for a proportion of triple-negative breast cancers. And I think that if we are able to identify those tumors that are deficient in DNA repair, well, for these tumors, PARP inhibitors represent an extraordinary potential opportunity. The problem is that only a proportion, most likely a small proportion of triple-negative breast cancers will be sensitive to PARP inhibitors. So it's quite clear that we need to identify other molecular targets for other subtypes of triple-negative breast cancer. For instance, one potential avenue of research that is emerging is the fact that some triple-negative breast cancers may be very sensitive to our immune system. And this is another way to go. The immune system and the host reaction against the tumor may be modulated, may be potentiated, for instance, cancer vaccines. And this is another way to explore. Once again, it's not going to work for all triple-negative breast cancers. It will work for that specific subtype of triple-negative breast cancers where the immune response is key for controlling and killing the tumor. Otherwise, it will be an ineffective strategy. So if you want, the PARP inhibitor trials that have been done so far have been giving a very good lesson. Don't consider triple-negative breast cancer as a unique entity. Consider different subtypes and target specifically the molecular alteration that you find in each different subtype.